have a long introduction. You do it?
I don't know if I can.
Yeah, that's a good point, Lynn. Then we'll make the candidate wear the microphone too.
I don't need a microphone in this room.
You know?
I'm very loud.
But you can be really loud with this. Okay, so you are an informed and loyal audience, so I'm going to make just a few brief remarks by way of introduction. I'm Steve Arnold, and I see the rest of Gwen's committee here is here, so that's good. It's traditional, I think, to start by saying when you first met the person you're introducing, but in Gwen's case, I could go back a generation and tell you when I first met her immediate ancestors, which was, I have to think about that. That would have been in the 70s when I met Bruce Burry, her father, and Jacqueline Wolfine, her mother, who at the time were graduate students at Berkeley. And I guess I'm a little grayer, and I was a postdoc then while they were graduate students. So anyway, so I knew both of her parents. And then we'll see the next thing, I moved to Corvallis, and looking back at your CV, I realized when we moved here, you were a high school student. And at any rate, so then switching over to Gwen, of course, she was an undergraduate at Southern Oregon University, and then a master's student at Western Washington University. You could see her arcing and spiraling back towards Corvallis. And for both her undergraduate work and her master's work, she worked on another cold stream indicator species, a scaphus, and she worked on their tadpoles and studied, in the first place, their feeding behavior and then their thermal response. So you can see, even as a master's student, she was equipping herself for her work on torrent salamanders here at OSU. And she's going to tell you all about, actually I don't know if you're going to talk are you going to talk about all of your chapters?
Yep, all four.
Yeah, all four chapters. So you'll see an integration of environmental physiology, lots of field work, lots of painful field work, and lots of enjoyable modeling.
I don't know if I'd call ARC enjoyable, but sure.
Eventually it gets enjoyable. At any rate, it's a pleasure to introduce Gwendolyn Wolfine Burry for her doctoral affairs. You don't want this.
I'm very loud. If anyone has trouble hearing me, let me know. But generally, loudness is more of a problem for me than an issue of being too quiet. So thank you very much, Steve, for that introduction. Yes, as you mentioned, Corvallis is apparently has very heavy gravity for me, and every time I go away and I get a degree somewhere else, and I end up back here. So I just sort of gave in to the gravitational pull and have done my PhD work here at OSU. So today I'm going to present a fairly complex way of looking at the effects of human impacts on a natural system. And the question first, of course, is why do we need to be looking at those human impacts? And the first reason is that there are just a whole lot of humans. There are currently 7.3 billion of us on the planet's surface, and we influence almost every process on the planet's surface at this point. And a lot of those changes are not positive. So we have human impacts negatively affecting both single species and entire ecosystems. And just a few examples, logging, fire, introduced species, hunting, and development. This is our lovely introduced bullfrog eating a duckling. And this is an aerial photo of the Cascades, and all of those paler patches are clear cuts. So climate change is based on human technology-produced gases. We'll go more into the details of this soon. But it produces increased temperature on the landscape. So this results in figures such as this one. This is from last year. This is June of last year. And the map shows warmer areas than average. So this is anomalies from the average temperature worldwide. So the warmer the color, the warmer the temperature on the surface of the planet. And climate change is already causing a lot of effects to natural systems. Range shifts, phenology shifts, extinctions of species, changes in disease prevalence. and tons of others that I'm not going to go into. But these effects are predicted to not only increase, but interact with all of those other human effects that I already described. So today we're concentrating on flowing freshwater ecosystems, and this includes both rivers and streams, and these are important not only to themselves as an ecosystem, but are widely connected to other systems on the planet, and are important to humans for the ecosystem services that they provide to us. Specifically, I look at headwaters in the Pacific Northwest, and headwaters are characterized as being quite small, extremely cold. They're very common on the landscape. They can drain up to 80% of a catchment is drained by the headwaters, and they also comprise the same amount of the river length, up to 80% of a river length. And as such, they're important to both the downstream ecosystem and to the surrounding forest. So if headwaters are important and imperiled, then how do we measure the function of this ecosystem? How do we look at whether it is working the way that it should be? One of many ways is to use indicator species. So indicator species are species that you go and look for on the landscape, and their either presence or absence or abundance tell you how that ecosystem is functioning. And each species is unique. Often use combinations of them, but you can also use single species, because each one responds differently to each type of stressor. So one of the important things that we need to know about an indicator species is their specific limits, because we're using them to look at an entire ecosystem, right? We don't just care about this animal, we're using them as an indicator, as the name says, of the whole function of the ecosystem. And for that reason, we need to understand what exactly they are responding to, so we understand why the patterns exist. In addition, when we do this, when we measure what's going on inside of an indicator species, we also not only get an understanding of limits, but we actually get to see those changes before they affect the entire species or ecosystem. The specific indicator species that I've been working on for the last 6 years is Reichitreiton variegatus. And these are a very small stream-side salamander. They're endemic to the Pacific Northwest. There are 4 species. I work on variegatus, which is the southernmost and extends into California. And these are headwater obligate. You only live in headwater ecosystems, nowhere else. Reichitreiton have been used as an indicator for decades. They were proposed by Hart Welsh as an indicator. And the populations are reduced and extirpated by a variety of different disturbances. It's well documented for things like logging and the silt caused by road building. In addition, we know that they have a low temperature tolerance, so a critical thermal maximum, this is where they lose function, at 28 C, which is the lowest of any amphibian that's been tested in North America. So I'm doing something kind of unique. I'm trying to forecast the function of a whole ecosystem, the whole headwater stream ecosystem, by looking at one indicator and what is going to happen to that indicator under different projections of climate change. So I have 4 sort of sub-questions or chapters, and these are, is the southern range edge of Reichitreiton variegatus temperature defined? At what temperature do adult Reichitreiton experience stress? At what temperature do larval Reichitreiton die? And does the range of Reichitreiton variegatus change under projections of climate change? So this is my orientation slide. We'll see this slide several times. And this sort of lays out my logic for each chapter and why each chapter is a part of the whole. So we've been looking at human impacts. A way to look at the impact of human impacts is indicator species. And then we need some information about that indicator species in order to do good modeling. And then my approach to trying to figure out that necessary information. So for all of my chapters, the human impact that we're looking at is water temperature, mainly caused by climate change, but also by other effects. The indicator species that I'm using is Reichitreiton variegatus, so these will be the same for every chapter. And then I will have different pieces of needed information and different approaches for each of my 4 chapters. So for chapter 1, notice the top part is going to be the same the whole time. So in this chapter, I'm looking to see, is the range thermally defined? And to do so, I did a field study looking at the temperature at the edge of the range. So why do we need to know this? We need to know this because there are lots of possible things that could define a species' edge. But when we are modeling, if we're assuming that climate change is going to change the range edge, then we're assuming that that edge is defined by temperature. It would be handy to know if that were true before we did all of this modeling. In addition, when we're using an indicator species, the assumption is normally that if you don't find them there, then something about the features of that area are unsuitable for them. So outside of their range should be unsuitable for this species. So my approach was to go to the edge of the range and measure temperature. This, of course, was a little bit more complex than it sounds. But we also wanted to measure other abiotic variables to make sure that something else wasn't defining that edge of the range. And so we went and measured a whole variety of things, both at the center of the range, where it should be very cold, at the very edge of the range, where it should be marginal, and then one sort of stream just outside of where they have ever been historically found, where it should be too hot if their range is defined by temperature. So to orient you to my study area and map, which we'll see many times over the course of the next hour, this is the border between California and Oregon. The lovely white star is Eureka, California, so Redding is over here. Mendocino County starts down at the bottom. So I had 28 sites. I had 8 sites in the core where I believed it to be very cold. These are the green ones. I had 10 sites as far east and south as Reichtriton have been found. Those are the yellow diamonds. And then I had 10 sites just outside the range. We went to an edge site and drove out until we found the first good headwater stream. It looked like it should have Reichtriton in it. And those were the extraliminal sites. And we visited all of these sites in 2011, 2012, and 2013. So the first thing we had to do was look and see if there were Reichtriton there. We did this using fairly classical amphibian survey techniques. We had a small net, put it in the stream, we turn over rocks, and the salamanders are washed into the net. We did a 3-person-hour search. So 3 different 1-person-hour searches. So a person hour, if there were 2 of us there, we searched for 30 minutes. If there were 3 of us there, we searched for 20 minutes. So it added up to 1 person hour. And we primarily searched in June because we believed we had the best chance of catching them in June, not in September when it was very hot at our sites. We also measured a wide variety of other abiotic variables. I'm not going to go through the entire list, but we looked at the substrate in the stream. We looked at width, we looked at slope, latitude and longitude, pool versus riffle, etc. And these were all variables that in the literature had, were indicative of being causative of Reichtriton being in the stream. However, our big question was temperature. So we put in these Magtech temperature loggers, and we used 2 in each stream because sometimes one of them dried up. So the second one was just a backup in the stream. And then we used 1 on a tree to measure air temperature. And these recorded every 15 minutes, which in hindsight was a terrible plan. So they recorded July 1st to September 1st for 3 summers with 3 loggers per creek at 28 creeks. And I then spent the next year trying to untangle my data sets. So, for example, so this is an example, don't get tied up in the details of this. This is one of our streams, this is Azalea Creek. And once we had all of this data, in addition to just looking at, for example, the maximum, we also looked at features like the relationship between air temperature and water temperature, and the slope and R-squared value of this line. We also looked at measures such as variability over the course of the day, variability over the course of the summer, and a whole bunch of other patterns which are on the next slide. These are all the measures of temperature that I used in this study. And if you notice, I now have way too many variables. So, in order to get something that a statistics program could handle without crashing my computer every 5 seconds, and that I could actually analyze in a meaningful way, I used a correlation matrix to reduce the number of variables in my analysis. Correlation matrix helps eliminate variables that appear to be measuring the same thing. So in my correlation matrix, I was fairly conservative. I dropped one variable from any pair of variables with an R value over 0.4. And I ended up with 13 variables that I then put into the next analysis. So the next analysis is a multivariate adaptive regression splines, which from now on I'll refer to as MARS. And this is a flexible, non-parametric, logistic way of looking at data that allowed me to see which of all of my many variables was important to defining the range edge of Reichertragen. And this gave me two variables that were important, and I could then use a general linear model to find the significance and effect size of those. So we did not find any Reichertragen outside of their range, 0 out of 10. We did find them in 80% of the sites inside their range, 9 of 10 of the edge streams, but 5 of 8 of the core streams, although later revisits, we did find them at more of these core streams. So if a site was occupied, we had a 72% chance of detecting it, and the probability that the site was occupied when we did not find them is only 2%, excuse me. So remember that the point of this is to define the range edge. So my dependent variable is not presence or absence of Reichertragen, but their historical range edge. So keep that in mind for the rest of the results. So this is the output from the MARS analysis. And the two variables that were important were the days over 13C, which I'll explain more in a moment, and the percent slope of the stream bed. And this is inside the range, and this is outside the range. And you can see that when there are more hours over a certain temperature, you find you are more likely to be outside the range of Reichertragen, and the opposite for slope. As it gets steeper, you are more likely to be inside the range. So a quick moment to explain what I mean by days over. So days over, or degree days, this is not the number of days that the stream was over 13C. This is cumulative hours over a set point, then expressed in days. So if we have our lovely example graph going up and down and up and down, and we lay a line across it, and we add up the number of hours, because these were recorded 15-minute increments, the number of hours above that line, you end up with huge numbers, and they're easier to look at if you reduce them to 24-hour days. So when I say that something was over X for 3 days, I mean over this cumulative number. So I also want to point out that 13C was slightly arbitrary. It wasn't only that streams were over 13C. There was a range of temperatures that streams inside and outside were different for. So from 11 to 15C, there's a different relationship for streams inside the range and streams outside the range. So streams inside the range are on average under each of these temperatures for 197 hours more. Okay. However, we're talking about tiny headwater streams. They're mostly groundwater fed. And at the ends of the distributions, there isn't that much temperature. Most small streams just don't get below 9C very often in the summer. So all of them go down to the same number at 9C, and also the same number over 17C. So what is determining the edge of Reichertragen? We came out with our two variables, slope and heat loading, or degree days. And the streams outside the range do seem to be at higher temperatures more often than those inside the range. So it does look like temperature has an effect on the edge of Reichertragen's range. As for slope, the steep parts of California, this is an elevation map, these steep parts of California extend beyond the range of Reichertragen. So I'm unsure if we were actually finding a real effect that Reichertragen primarily live in steeper slopes and that it is flatter outside the range, or if, because our sample size was not super huge, if we just happened to pick a couple of really flat streams outside of their range. This is important because this has implications for human actions that increase stream temperature. Not just climate change, but also urbanization and logging both distinctly increase stream temperature. And if Rhycotriton are only living in areas with a lower heat load, this would impact where they can live in the future, and also impact our ability to model on range edge. So having discussed range edge, we're now going to move on to some of the other things that I needed for this study. So the next thing that I needed was some more information on temperature tolerance. We only had critical thermal maximum, which is the temperature at which an animal dies immediately when it is brought up to that temperature fairly rapidly. So in this experiment, I'm looking at the stress levels in adult Rhycotriton. Why do we need that? First of all, this is a causative reason for decline. We're looking at what in the body is actually making them not appear on the landscape. And this also responds before that whole population and species level. So specifically, I looked at corticosterone, or CORT. This is a hormone that rises with stress. It's highly conserved, so it's fairly comparable, and it's relatively easy to measure from a variety of animal parts: blood, hair, scales, mucus, feces, urine, and in aquatic species, body wash. However, there were some challenges to this. Rhycotriton are very little. Getting a blood sample from them can be kind of challenging. This has never before been measured in this species. No one has ever measured their levels of CORT in this species. And just to make our lives even more interesting, we decided that we really needed to use realistic treatments, not a steady temperature, but a varying temperature as we see in nature. So, Kaylee and I learned a whole lot of plumbing and wiring. So we constructed these complex baths. I was unable to afford commercial benchtop chillers, and luckily one of my former advisors was able to point me towards a chiller setup that I could afford. And we ended up constructing these water baths that had switches so they turned on and off during the day and at night. They had both heaters and coolers, and we had switches that controlled the heaters and chillers so that they stayed within 0.3 C of whatever temperature we set them to. And using all of our complicated things together, we managed to make these water baths that fluctuated very similarly to the ones that we saw to the fluctuations that we actually saw in our field study. So we set them based on that field data. So the amplitude was set based on our field data, and so were the temperatures. So our lowest temperature was 12 to 15, and then 15 to 18, 18 to 21, and 23 to 27. So this is our sort of diagram of the whole experiment. We took adult Rhycotriton variegators, we put each one in a little box. We put 5 boxes in our big water bath. The water bath started off cold, went up to our daily maximum, got cold again, so a 24-hour treatment, and then we measured stress level. And in this case, we measured stress level from a blood sample. So we measured the stress level by taking a very tiny blood sample from the upper leg of the Rhycotriton. There's a visible artery there. We made a tiny pinprick through the skin, and we're able to gather the blood in a small capillary tube. And then we sent the plasma up to OHSU, and they analyzed the levels of CORT in the plasma via enzyme immunoassay. That's pointer, not the clicker. Then we did the entire experiment again. We got new Rhycotriton, we put them in boxes, we put them in our treatment tanks, we ran it from cold to warm to cold, and this time we measured stress levels from both blood and water. How does that work? So, take your salamander, put it in an appropriate vessel, with water, wait 30 minutes, take the salamander out, take the same blood sample we took before, measure CORT from the plasma, and then additionally filter the water and analyze that for CORT. Again, by EIA at OHSU. So results. This is the first experiment where we only looked at blood levels of CORT. So we had a doubling of CORT from here to these 2 treatments. And then a 10-fold increase up at this treatment. This one is significantly different from these 3. These 3 are not significantly different from one another. and overall p-value is highly significant. So remember, we did the entire thing again. So we have blood results the second time. This time we saw much more variation. We still saw, though, the same pattern where we had a very large increase here and smaller increases below. And none of these were significantly different from another. The good part about this, however, is that the water looks very much like the blood results. So that is one blood and one water sample from each individual salamander. And we found a strong correlation between the water and blood quart in each animal. Had an R-squared value of 0.67. And for each individual, these are the paired measures. So this is an individual salamander with water and blood for each one. And you can see that there is quite a tight association for each individual. So for stress, we saw a doubling of quart over temperatures where they're not found. So ricotryton are not found in nature. If you measure the water temperature right when you're looking, water temperature is over 15, you don't find ricotryton. When we increase the temperature over 15, we saw a doubling in quart. A doubling in quart of the same magnitude that we saw has been significant in a bunch of other studies, but not in ours. And then we saw a big increase, very significant, in quart near their critical thermal maximum. So this is a different stress response. So the lower stress response, the doubling, is the sort of emergency life history, something very bad is happening to me response. And the really dramatic change near their immediate death point is like a final effort to survive. So this is similar to other amphibian responses in the literature. The baseline quart measurement is very close. The doubling of quart at survivable field stress and the large increase in quart near the lethal treatment areas have all been seen in other amphibian species. So this is starting to explain why we're seeing this pattern on the landscape. We're seeing stress at higher temperatures, and especially at the temperatures that we know are boundaries for them. In addition, this chapter is important because we made a lot of methods advancements. The non-lethally blood sampling technique was something that I did in my master's for the first time, but which has not been published. And we also were able to make affordable daily cycling temperature treatments, which are important not only to us, but to lots of other people. And this is the first time that water quart has been used on a West Coast amphibian. I realize that it's very common in the fish literature. Hasn't been done as much for amphibians. And that water quart really has a lot of possibilities for the future. So you only need to test the association between blood and water quart once for each species. And from then on, you can just take the water quart samples. And this means that you can take field baselines, you can do repeat measures on individuals, do no harm sampling, and even take multiple hormones from a single sample. So now that we've covered adult stress, the next thing we need to look at is larval limits. And in this case, this is survivability. So why is it important for us to look at multiple life stages? So far, we've only been looking essentially at adults. However, adults are not necessarily what's limiting a species on the landscape. And any life stage is, if the abiotic factors are above the tolerance of any life stage, that limits the range of a species. And adults are often, not always, but often not the most sensitive life stage. So again, we used realistic cycling treatment temperatures so that our results would be applicable to field conditions, and because animals actually respond differently to cycling temperatures than they do sometimes to a steady temperature. So, same plan as before, large bins, heaters, chillers, all of the fancy things we could wire and plumb together. This time we have more treatment temperatures, and we ran it not for 24 hours, but for 21 days, or until the animals had all died in the treatment. And we used 5 larval salamanders per treatment. So, interestingly to me, this was not what I expected to happen, all of my salamanders survived up until a high of 23C. At 23C, only 1 died. at a high, a daily maximum of 25 C, 4 of 5 died, but the fourth one didn't die until day 15. And then when the temperature was increased to a daily maximum of 27 C, they were all dead on day 3. So the LT50, the value at which you would expect half of your salamanders to survive your 21-day treatment, is 24 C. This is interesting. They survive right up almost to their CTM. So a critical thermal maximum means you put them in that temperature, they're done immediately. And these animals were surviving daily fluctuations for about 3 hours up to almost that temperature for 21 days. However, some of these responses are also seen in cold water fish. I read one paper which hypothesized that this was an adaptation for cold water species, that though they are mostly adapted to very cool, they can occasionally survive a few peaks of temperatures up near their fatal temperature. So larval survival is not what's limiting Rhypotridon to areas under 15 C. Are eggs limiting them? This is possible. I attempted to have this experiment, but cord leak can be vindictive and melted down our cold room. And so I had an experiment set up with the eggs, but I had no control for it. Half of those eggs died when our cold room went up to 18. This is a similar response to salmonids, which also shows that this species is probably a good indicator for not only for conditions in headwaters, but also for salmonids in downstream areas. So now that we've talked about larval survival, we've built up a whole lot of those components that I was talking about earlier that you needed to have before you started doing the modeling for where Rhypotridon can live in the future. So the first thing is, why do we need those estimates? Why are we looking at what happens to a species in the future? And first of all, this is our pressing reason, is that we understand that climate change is already affecting species, and that it exacerbates all those other human impacts that are already affecting the landscape. And as I mentioned earlier, this is a sort of a new way of looking at a whole ecosystem dysfunction. So here we're looking at a single indicator, we're projecting what's going to happen with that indicator, and using that to say what is going to still be functioning in this entire ecosystem. And it's important to do that for headwaters because they are not only essential to themselves as an ecosystem and downstream areas, but also to the upland forest that surrounds them, and of course to humans who rely on them. So now we need to get a little bit into the details of how we do this modeling. So the components that you need of modeling a range shift. So first you need some climate change scenarios. We'll get to those in a moment, but they need to be specific to the area that you're looking at, both in the present and in the future. You need specific responses of your species to temperature. You need range determinants, what is determining the edge of their range. And then you need some computer methods of estimating that range, both currently and in the future. So climate change is radiative forcing from anthropogenic, mostly CO2 emissions, although there are other gases that certainly contribute to climate change. Now, future human actions are unknown, so there's a wide variety of what we project is going to happen. And in this study, we're using the most recent scenarios from the Intergovernmental Panel on Climate Change, the IPCC, which are called representative concentration pathways. So representative concentration pathways, there's 4 sort of possibilities of what is going to happen over the next 100 years. So these possibilities are in watts per meter squared, but they are an estimate of how much CO2 is going to be in the atmosphere to cause this much change in temperature. And there is error around each of them. So we have the most severe, which is the 8.5, moderate at 6, another moderate at 4.5, and the lowest at 2.6. And notice that these don't all just increase linearly. So each one of them has a different pattern of increase. So 8.6 goes up quite steeply, whereas 4.5 and 6 are more complex, and 2.6 actually sort of peaks and then decreases a little bit. So in addition to the variability in representative concentration pathways, we also have general circulation models. So a general circulation model is a global scale model of what increased CO2 is going to mean at a ecosystem, well, at a more local level. What is that increase in CO2 going to mean to ocean temperature, to glaciers, to ocean acidity, to precipitation, et cetera, et cetera. And each of these models is unique, and there are a lot, dozens and dozens of them. So the question is, how do you choose which ones to use? Generally, you look at how the model performs in the past related to the present, and then use the ones that have worked well in the past and present, and assume that those ones will also work well in the future. Luckily for me, I didn't have to do that analysis, because there is this handy review for Pacific Northwest projections for hydrologic systems. We were also limited by which ones we could get for free online. So we used a wide variety of them. We're trying to look at variation. But as you can see, for some of them, we could only get one representative concentration pathway, where for others, we could get all four. Don't try to, this is here to show the great variability. I'm trying to memorize the bottom of this slide. So in this study, we used all four representative concentration pathways, or as many as we could get for each general circulation model. We used 6 general circulation models, and we did all of these for both current, 2050, and 2070, which is 37 combinations. So 37 either presents or estimates of what the future is going to look like. In addition to an estimate of sort of what the temperature is going to look like in the future, we also needed a way of looking at how that's going to affect our salamanders. So there's 2 sort of big umbrella ways to do that. And one of them is correlation, and the other one is trying to use a causative model. So a correlation model, you look at climate and other abiotic variables, and this is usually called an envelope model. We'll go more into this in a second. Or you can use a causative model, where you look at the limits of that species and say, okay, beyond this limit, they cannot live in this specific place. And that's usually called a mechanistic model. So we had our 37 combinations of what the future might look like, and then we used 2 correlation models and 1 mechanistic model. So how does that correlation method work? So essentially what we use is we use the current occurrence of the species and then a bunch of layers of abiotic data, and we sort of stack those layers on top of each other. So here we have a mythical species. These are all the places that the mythical species lives on the map. And then over this, we're going to put some abiotic variables. So here we have an area that is cold and an area that is hot. We have some elevation, and we have some precipitation. And if we sort of drill down through each of those layers, we see that our animals only live at a low elevation in the warm area, but they live across our 2 measures of precipitation. So we can then use that to set boundaries of where we expect them to live on the landscape in the future. There is a lovely program called MaxEnt, which does this, and it uses a maximum entropy approach. So it chooses 10,000 random points from the background and compares them to the points that you gave it to see sort of a degree of randomness in what is going on and whether your projections fit with randomness or fit with a pattern. And each run of this is unique. So for our MaxEnt inputs, we used a bunch of climate layers, which I will show in a moment. We also used the point localities downloaded from VertNet. So the same study area that we were using before in Northern California. And we downloaded 116 localities representing 450 individuals. We ran this 25 times, and we used the mean, and then MaxEnt provides a jackknife of variable importance. And again, this is the same map as before, so Eureka is here, Redding is over here, and this is the border between California and Oregon. So the first time that we ran this correlation model, we used 14 variables. And this is sort of the standard way of using MaxEnt. You look for a bunch of variables that you think are going to be important to your species. And again, we know some of the variables that are important to Rechytriton from the literature and from our own other studies. And then you put all of them in and you see which ones are the most important to that jackknife. So we use mostly measurements of temperature, but we also included 2 of precipitation, and that includes the seasonality, both of precipitation and of temperature. Oh, and in addition, we used elevation and slope. So, then we ran MaxEnt again. And this time when we ran MaxEnt, the only things that we put into this model were elevation, slope, and the maximum of the yearly temperature. 2 of these are static, only one of them changes with projections of the future. So these result in output maps. You get maps with a range of values, and one of these standardized ways of dealing with all of that data is to classify it. So you say the top quarter of the data represents the best area for Rechytriton to live. And then stepping down, then you have excellent, fair, marginal, and not suitable habitat. Each one is a quartile. And this was classified from the current data, and those same breakpoints were used in the future. In addition to that correlation model, we also used a mechanistic model. So a mechanistic model says, here is my landscape of temperature. Where in this landscape can my species live, based on what I know about their tolerance? So we used that data from chapter 1 on air and water temperature, and we made a linear regression. We put in a whole bunch of those variables that we studied, and we said, what is important for the yearly maximum water temperature of a headwater stream? And our regression said the important things were elevation, slope, and air temperature, so that's what we used. Again, we classified, but in this case we classified on the boundaries that we know for Rechytriton. So Rechytriton in the literature are often not found below 8C. This was generally not a problem, as only the very top of Mount Shasta is apparently too cold. Then we had the temperatures at which they're almost always found in the field, temperatures where they're usually found. Above 15, we do not find them, so we classified that as marginal. And then above 17, we classified as more stress, as 17 is a cutoff where Dr. Welsh has seen whole body measures of stress where they lose function. That made too many maps. So we had 37 presents and futures times 3. I can't really show you that. So we're going to show you some averages, because part of the point of this chapter is to not only look at one projection, but to look at what a whole bunch of projections show for the future. So we're going to look at averages across a representative concentration pathway at each time period, and then a lot of this is going to be in percent of the map area in a certain category. So first of all, how did our model fit? How did it work? MaxEnt says that it worked really well, actually. So AUC is area under the curve. It measures how well the model performs in comparison to a random selection of points. Remember, this is how MaxEnt looks at how well your model fits. And the line here is random, and this is our line. So if it were random, the value would be 0.5. Over 0.8 is a good fit. We had 2 runs, the multivariate and the yearly maximum. So as far as fit, we're doing well. We did a jackknife, so remember the first time we ran MaxEnt, the multivariate, we had 14 variables, but only 5 of them were over 10% of mean contribution to the model. So 5 variables explain more than 10% of what happened in the model. And those 5 were precipitation, altitude, seasonality of temperature and precipitation, and annual mean temperature. We also ran a jackknife on all of the variables that we put into the yearly maximum MaxEnt to show what their percent contribution to the model was. The most important 51% of the model was from altitude alone, and then maximum temperature, and then slope. So how does that fit to where they actually live? Which of these projection maps most closely fits where we know Ricotridon live on the landscape? So this is our multivariable maxent, our yearly maximum maxent, and then the mechanistic model. And as far as colors, red is not suitable, yellow is marginal, light green is fair, and dark green is excellent habitat for Ricotridon. So the multivariate maxent is a closer fit than any of the others. The other two are much more widely overestimating where we would expect to find Ricotridon. Since in chapter 1 we searched all out here and didn't find any, we're fairly confident that when we say they aren't there, they're not there. So the percent area classified into each category. So what percent of this map is red? Notice that the multivariable maxent starts out with a whole lot of the map red to begin with. Over 65% of the map is already out of bounds. We did this slightly on purpose, as when you are modeling, it's good to show the whole range, so you see very cold to very warm. And it also is interesting to compare to our other two, as the yearly maximum maxent had less of the map as not suitable, and the mechanistic actually had most of the map as fair habitat. Habitat change over time, this is roughly what we would expect. So this is the percent of the map classified as excellent. So how much of that map is dark green? And the percent of the map falls over time. As we go into the future, under any projection of climate change, there is less area on the map that is excellent habitat for Ricotridon. So now we're looking across representative concentration pathways. So this is the map for current. Then we have the lowest increase in CO2, working our way up to the highest increase in CO2. And this is a mean of all of the different general circulation models that we ran. So between 3 and 6 general circulation models for each map, past current. So as you can see, we have quite the decrease in good area for Ricotridon as we get higher and higher CO2. Comparing to the yearly maximum maxent with the same setup, again a decrease in a slightly different pattern. And for the mechanistic model, again a decrease, again in a different pattern. So comparing all three, we see the most drastic decrease in the multivariate, with more of a decrease centering on the coast in the yearly maximum, and more mixed in the mechanistic model. So generally, over time, we have less excellent habitat. Generally, as we project into the future, the more CO2 there is in the atmosphere, the less excellent habitat there are for Ricotridon. However, of course, we are multiplying uncertainties here. There was error in every single one of those models that we put in to our model. And so this is definitely impacts what we do when we're doing this type of modeling, which is why the literature advises to use the multi-model approach as I did. Sometimes if you project with one model, one type of modeling, then you get a projection and you are relatively confident in it, but these may show very different patterns if you use different models. However, adding sort of infinitely more models does not always increase how good your modeling effort is. Sometimes it just adds noise. So where your cutoff should be is currently unclear. I would say, and the literature supports me, that a combination model is really needed here. The mechanistic model misclassifies high elevation areas. It thinks that this big, dry, high desert plateau over here would be excellent for Ricotridon because it's cold. It also thinks that these dry ridges would be really great for Ricotridon. They're oak woodland. There's no water there. So this is obviously a failing in a mechanistic model, and did not happen in the correlative model, because the correlation model said, oh, they don't live on dry ridges and in high desert, we'll not put them there. So this is an argument that we do need to combine those, but the correlation models don't do things like take into account things that the mechanistic model does, such as that groundwater may not change as fast as air temperature changes. And that this is a causative model. This gives us a reason why they would be there or not there. So more information generally gives more accuracy, which was the point of this study. I was looking for the determinants of range edge, which looks like it's involved with heat load, the temperature tolerance of adults and larvae, and then modeling potential range shifts under climate change and the importance of looking at variability when you do so. We still need some. We still don't have a real solid causative reason why we don't find them over 15 C. We don't know the tolerance of eggs. Eggs are certainly the limiting life stage in many other species. And we don't have a complex understanding of the air-water temperature relationship. I used a fairly simplistic model here. I was using it in ARC, and I was unable to incorporate a lot of the more complex measures. We also don't know how much groundwater temperature is going to change with air temperature changes, and whether Reichitreiten are really only using that surface water, which is mostly what we're testing, or whether they're using water that's flowing underground, which has other capabilities. However, it does appear that increased temperature leads to decreased Reichitreiten. And having an unknown mechanism doesn't invalidate that observation. We completely log an area and it becomes very warm. There's no Reichitreiten there. When we go outside of their range and we measure and it's very warm, there's no Reichitreiten there. So just because we don't know why they're not there doesn't mean they aren't still a good indicator. They're definitely highly sensitive to temperature and in many ways very equivalent to a lot of the cold water fish in the Pacific Northwest. And both populations and individuals respond negatively to increased temperature. So this was a unique approach because all of those big maps that show that it's red all across the landscape, that's not only that Reichitreiten are going to have difficulty living there, as an indication that the whole headwater stream ecosystem is going to be in trouble in those areas. Our modeling shows a drastic decrease. So if we want to look at these headwater systems, we should realize that the next 50 years may be not a good condition for many of them. We modeled only climate change. This did not include any other effects. This did not include increased temperature from logging. This did not include any other human effects on the landscape. This was solely looking at the effects of climate change. However, human actions are not set over that time frame. In the next 60 years, there are a great many things that could happen that could decrease the CO2 in the atmosphere, that could mean that we would only go as far as one of the much more moderate projections of climate change. So this is all very much dependent on what we do in the next 60 years. I have a great many people to thank for my overly giant PhD, EPA star, for funding me for 3 years and giving me some really amazing opportunities. Dr. Hauk's NSF grant, my advisor, and my committee. Dr. Olson for loaning me loggers, without which I would not have been able to do that field study. The Integrative Biology Department, the office staff especially have always been amazing. Angelo Coast Range Reserve, the California State Parks, and the Hoopa Valley Tribe for letting me tromp around in their streams. My field crew, without which I would not have done any of that work. Primarily my father, Dr. Bruce Berry, for driving me around in the Big Red Track. Big Red Track deserves a thank you of its own, and providing me with pocket money for beer and pizza, which is essential for paying off your field crew. Christy Baggett and Kaylee Doughton, who rolled an amazing number of rocks and did miracles in the lab. Jason Brecken, who the first year I think caught about 2/3 of the salamanders that we encountered. All of my undergrads who were not only happy to be in the field when it was sunny and we were catching tons of stuff, but were still happy to be in the field when it was pouring rain and we hadn't caught anything in 3 days. So they're all amazing. My office mates, Lisa and Lana, of course, who somehow, things like this are always happening if Lisa and Lana are around. Miriam and Alyssa for coming to the office and buying into all of my craziness and tomatoes and not like minding any of it. Sarah and Yosef and Karen and Adam, who were always there for me to ask questions of. The other people who made it all possible, Dr. Michael Parker and Dr. Yvonne Erzmindi, who always had an open door when I was totally stuck, had no idea what I was going to do, and let me bother them quite intensively and more than was required. Joseph Butterworth for infinite patience with me and providing me with buckets of kittens when I was going to have a meltdown. Has happened more than once. Piper and Valerie and Sally and Militia Small for photos, and also Christy and Kaylee and various other people whose photos I lifted from Facebook. All right, any questions?
All right, great. Committee members will get a chance in the closed door session, so we're going to give priority. If you've got a burning question, yeah, we're going to give priority to other people to ask questions now.
So it seems to me like there's this really interesting, not necessarily disconnect, but there's this really interesting pattern between where in the lab we found that court doesn't increase that much until you get to critical thermal maximum, and then the larval survival is fine until you hit really, really high temperatures, and your mechanistic models overestimate the species range, but you don't find them outside this really narrow range of temperature. So are there other factors you think that could be setting the range edge other than temperature? Because it seems like it could be dispersal or species interactions or some other abiotic factor.
Right. So dispersal is a possibility because especially in more northern areas, this species may still be spreading out of Pleistocene refugia. However, we don't find them over 15 C, right? So no matter where you go, you're not finding them over 15 C. And down in California, those areas are getting over 15 C much more often, right, than inside the range. Remember that? I promised I would not draw in the air. That I would go back to slides. So I think it's probably eggs. So other species that live in very cold areas have eggs that die at very low temperatures, like 18 C. And half of ours died at 18 C. So I think that population-wise, it's probably the eggs, but the cold room stymied that effort.
So is there any evidence for something else that could be correlated with temperature, though, that could be excluding them from those habitats? Something like a disease or a predator or...
So it may be, so when we find a lot of Reichtriton, we find fewer Dicamptodon, which may be, oh, sorry. All right, computer says it's done. So it may be their main predator. Well, we think, anyway. This is complicated. The Dicamptodon, which is a large salamander, strikes on movement and bites them, but half the time it then spits them out again. So we don't know if they're noxious, we don't know if the dicamps are eating them all, we don't know if it's competition, because the diet, especially the small dicamps and the diet of the Reichtriton are identical. So it may be that there's not enough prey items, and if there's too many dicamps which have a larger range and a larger tolerance, that there's less Reichtriton. However, there's dicamps everywhere. So they can't be excluding them, because they're not only throughout their entire range, but have a larger range than the Reichtriton have. So I'm not sure that it's a predator. I don't think it's a disease. I think that their ability to get to a new stream is excessively limited. They're not only stupidly sensitive to temperature, they will, if you hold them in your hand, they will die. They're also really sensitive to desiccation. We had problems in our lab if one of them would get out and just get onto the floor of a cold room. They would desiccate and die in hours. So their ability to disperse across the landscape, even on like the geologic time scale, is not super fast. However, That being said, some of the other sort of semi-terrestrial salamanders in the area have actually contracted some because Northern California was sort of more like Oregon or Southern Washington. So some of the other species actually had a larger range and have now contracted back based on, again, some kind of abiotic determinant. And it seems like temperature is at least involved. I'm not sure that it's the be-all and end-all, especially like when I do those correlative models, and the top variable is precipitation. So I'm like, we should look at precipitation here. But that's not something that I could do from a mechanistic standpoint at this juncture. So, that answer? Yeah.
I have a question about the maximum modeling and the amount of time after that went into the BirdNet samples that we were using.
BirdNet's super easy.
Well, so how time average were those? Because do they go all the way back to 1890 or were they limited to the last decade?
So the earliest ones that I found are, let's say 1950s. I didn't check very closely to make sure that there weren't any earlier ones in there, but it's almost all from the Berkeley collection. And there weren't like a lot of serious collecting of Rhycotriton until geologically, relatively recently. And I did not include that variation in like the Maxent to say, okay, these were from 1950, are they in a different pattern than the ones that were collected like in 2010?
Yeah, because the temperature could change a lot between 1950 and 2010. So that might be worth doing, but it also might give you the chance to actually try time casting. So you have your three model approaches, and you could actually well, you have to be careful about how you build the model, but if you could then practice or see how well the models do with time casting and predicting where you know those localities were for the past, then it might help you narrow down which approximation would be more.
I would love to do that because I also really want to test that, like, was their range actually larger and is currently contracting? Or were they in a refugia and are now, like, expanding out? Because the northern ones definitely are. There's contact areas between the northern species. The southern one, I'm, like, not as convinced. Some of the genetic data doesn't, I'm unclear about what's going on with the southern group. I did a question. Yeah.
Fantastic presentation. You have a great presentation skills and values. So I'm wondering if you could clarify the role of precipitation seasonality for me. Obviously, as you know, climate change is going to have a huge impact on snow to how much precipitation falls with snow and how much is rainfall through the headwater, groundwater, headwater streams. So how is that going to factor into all of this?
So the seasonality actually is a really cool pattern. This is dead. I'm drawing on the chalkboard. Okay, so you have Oregon, Washington border, nose, right? So mostly you're finding Rhycotriton in areas that are right in the redwoods belt. So there's almost no seasonality of anything. There's almost no seasonality of temperature. There's almost no seasonality of precipitation. I mean, there certainly are heavier rainfalls in the winter, but certainly nothing like this really dry belt over here, where you're getting like snowpack in the winter and then a completely dry summer with almost no precipitation at all. And this is the pattern, I believe, that's driving that difference in the association, the like at what seasonality do you find Rhycotriton? Because it's showing that you find Rhycotriton where you have less seasonality. The trick to that is that there are some projections, not all, but some projections that show that that entire system, the like suck cold air in from the coast, redwoods like precipitate the fog onto the ground, et cetera, is going to get disrupted by climate change. So if that happens, I think this coastal population is in big trouble. So yeah, there's a chapter 5, didn't get done. Data got done, didn't get written. And in it, I did some analysis of which of these areas, so which ones are going to be refuges and which ones are just going to get wiped out. And there's two types of refuges. There's one that's the seasonality refuge, where as long as that holds with the essentially no seasonality, that's a refuge. The other one is we have some streams where it's really hot in the summer, like really hot. and really cold in the winter, and the groundwater temperature doesn't change. It is like not even a degree the entire time. It's just there, it's spring, comes out of the ground, it's like the length of this room. We catch more record in there than we catch anywhere else. And as long as there is a flow of groundwater at all, I'm pretty sure that place is just doing its thing. I mean, it will be 105 air temperature, 11.2. Like, oh, it's 32 air temperature, 11.2. No difference, doesn't care. They're record. All right, you want to keep going?
I think we probably should call it quits and move to the next phase. All right.